Okay, so our next speaker is Robert Rhoads. Robert comes from Louisiana State University Health Sciences at Shreveport, Louisiana. He's done a great body of work in the area of protein synthesis, and he's going to talk to us today about translation initiation. Robert Rhoads.
I'm talking about something that is sort of my career. This protein 4E is one that I discovered. My first graduate student discovered this protein, and then we were the first to clone it and have done a lot of things with it. But rather than a historical treatment, which she did, of cytochrome P450, I'm going to do sort of 6 short vignettes of where the research is right now, and just 6 stories that we are pursuing, and kind of represent the different types of approaches that we're taking. The general area is initiation of protein synthesis. Complexes are built up from the small ribosomal subunit, the initiator tRNA, the messenger RNA joins, the large subunit joins, and they're all catalyzed by initiation factors. This has the most factors. It's also the most complex of the steps and the most highly regulated, which is where messenger RNA is put onto this initiation complex. The polypeptides that are involved in mRNA recruitment include eIF4E, that also is involved in methionine tRNA recruitment. 4A is a helicase that's involved in unwinding messenger RNA structure, secondary structure that allows the ribosome to move. 4B assists 4A in its helicase action, as does 4H. 4E is the protein I'll primarily talk about today, and it's a cap binding protein. And 4G is the binding partner for 4E, but it's also the binding partner for everything else on this slide. It holds them all together, including the poly A binding protein. And this is how they all come together on the 48S initiation complex. So 4G has binding sites for all of these proteins right here, and it's sort of the glue that holds it all together. This interaction is critical because this is where messenger RNA is recruited to the translational machinery. That is the cap4E and the 4E4G interaction. So what's a cap? This is the mRNA chain. The last nucleotide is reversed 5' to 5' with a triphosphate, and it has a methyl group on it. So this is the signature for messenger RNA. And by virtue of that nucleotide, it binds very specifically to 4E. So there's a very narrow cap binding pocket, pi-pi interactions, hydrogen bonding, and then ionic interactions. So this is the purine ring, and then the ribose, and then those are the phosphates of the cap. So it's reasonable to ask, why is 4E interesting? There's several reasons for that. First of all, this affects the spectrum of messages that are translated. And this is because you can see that this recruits the unwinding machinery through the RNA helicase 4A, the original dead box protein. And so messages that have high secondary structure have a greater requirement for this unwinding machinery, and those with virtually no secondary structure almost don't need it. And so by changing 4E levels in activity, you change the spectrum of messages that are translated, and therefore the spectrum of proteins into a cell. Because of this, overexpression of 4E can greatly affect cell physiology and take a normal cell to an oncogenic cell. If you underexpress it by RNAi or other means to downregulate by inhibitors of 4E, you can reverse the transform phenotype. And for this reason, 4E is very relevant to clinical cancer. There's a large number of clinicians that study 4E as a marker for cancer stage, and it's also a target for intervention because downregulating 4E reverses the phenotype. And then the regulation of this protein is varied and complex. I'll be talking about one aspect of that. So besides all of those translational aspects of 4E, it's also involved in silencing of mRNAs by binding to proteins specific for 3' UTR elements. It's involved in patterning of protein synthesis throughout the cell and through early embryos. It's involved in mRNA transport out of the nucleus to the cytosol. And it is an element in stability of messenger RNA because it competes with decapping enzymes for degradation of message. So these are my, my 6 vignettes. I would like to first talk about how changes in 4E can affect this critical interaction between the cap and the protein. So looking at the effect of phosphorylation of 4E, and then what happens when 4G binds to 4E? How does that change cap interactions? Then turning the tables, how does changing the mRNA cap affect this interaction? I want to talk about something called anti-reverse cap analogs and also analogs that can prevent mRNA degradation. And then in a whole organism, we have been studying 4Es, and I'll talk about one particular one, a family member of 4E in C. elegans that has specificity for one class of messenger RNAs, and then how we've been using bioinformatics to study how this process occurs. So the first story, these are my collaborators, is the effect of phosphorylation. So here's a brief history of phosphorylation of 4E. We discovered it back in 1986 and didn't really know what it was good for, except that we saw that under conditions where protein synthesis was going rapidly, phosphorylation of 4E was very active, in particular incorporation of P32 into 4E. We determined the phosphorylation site in '94, as did another lab. The kinase was discovered later, and then, as you saw on an earlier slide, this kinase MINC binds to 4G. So it's a dedicated kinase that's right in the translation machinery. Some SPR experiments, surface plasmon resonance experiments, showed that phosphorylation increases the off rate for the cap from the protein, but does not affect the on rate. I use the word claim because it turns out not to be true. And then whole animal experiments indicate that phosphorylation has something to do with normal growth and development. This is all the positive aspects of it. The negatives are that a lot of papers have been published that show that phosphorylation of 4E does nothing. Overexpressing MINC or knocking out MINC doesn't change anything. So it's one of the most controversial elements in protein synthesis. We have almost the same number of papers saying it's really important as the number of papers saying that it's not important at all. So we first went into this to study what is the effect of phosphorylation of 4E on cap binding, and we wanted to use a stop flow kinetic analysis for this. Take advantage, if you remember the tryptophans in the initial slide of the cap binding pocket, when a cap analog binds to 4E, it quenches the fluorescence. And so this is titration of 4E with increasing concentrations of a cap analog, and that is a measure of cap binding. So we can use this in a stop flow setup where we have a cap analog in one syringe and 4E in another, and then follow the kinetics of quenching of fluorescence. So this gives us single exponential fits to the data, which indicates it's a simple one-step reaction. The previous data said that it was a two-step reaction, but we have an explanation for why they got that result. When you take the observed rate constants and plot them against cap analog according to this equation, you can get on and off rates. We can see that KCl strongly influences this, that the kinetics are inhibited by higher and higher salt. But if you now compare the unphosphorylated to the phosphorylated form of the protein, you can see that the on rate is about 2.5 times lower when the protein is phosphorylated than in its absence, but there's no effect on the off rate. So we interpret these results in the placement of where that phosphorylation is. So this is the cap binding pocket and an incoming cap analog. The serine 209 puts a phosphate group right at the entrance to the cap binding pocket. So we think that there's electrostatic repulsion of an incoming cap, and that's why we see inhibition at that point. The net result of taking the on and off rates is that you've lowered the affinity of the cap when you phosphorylate it, which again is confusing initially to us because this is something that correlates well with the rate of protein synthesis. When protein synthesis is going fast, 4E is phosphorylated. So why would you expect an inhibition? We think that it actually has to do with a cyclical phosphorylation, dephosphorylation that occurs with every round of initiation. You have to bind a cap, but before the initiation complex can move on down looking for the AUG, you have to unbind a cap. And so we think that's why MINC is located on 4G. You bound the cap, you've recruited everything, you now phosphorylate 4E to make it let go. So, 4G. There's a long history of the effect of 4G on cap binding. And I've summarized that history. It's widely believed that 4G allosterically changes 4G, allosterically changes 4E to increase its affinity for the cap. So this started with a paper from Nachum Sonnenberg in '97, where he said it dramatically increases the binding. Followed up by a paper by John McCarthy in Embo Journal with the yeast proteins. Another paper from McCarthy in 2000. Gerhard Wagner contributed some NMR studies for this that says that there's this conformational coupling between the two. Another paper by Wagner and McCarthy in JMB. Dixie Goss got involved with some fluorescent cap analogs. And then with the plant 4Es, this was also found to be that in all these cases, binding of 4G would increase the affinity of 4E. So we had completed this kinetic study on the phosphorylation, and we wanted to know how 4G affected these kinetic constants. So we expressed now 4G is a very big molecule, 1,600 amino acids. It cannot be made in bacteria. You can make little pieces of it. And so we made the 4E binding domain, which is about 100 amino acids long, expressed that and showed that it did in fact bind 4E. So this is a cap column, an M7G column. 4E alone binds to the column. 4G alone does not bind to the column, but when you put 4E and 4G together, you can retain the 4G stoichiometrically. So we get a one-to-one binding between the two. Now we did stop flow experiments, but this time in the presence of 4G, and we saw exactly the same kinetics. So the open circles are with 4G and the closed circles are without 4G. No difference in the kinetics at all, despite all of those fancy papers in Embo Journal and Cell and all like that. So this gave us pause. Why are we getting a different result? So we wanted to take a totally different approach to confirm this. For a simple one-step binding, the ratio of the on and off rate gives you the equilibrium constant. And so we found that K2 is equal to K3. So in this scheme, so if you take 4E and a cap and look at the binding, the on rate to form that complex, it was the same as in the presence of 4G. So K2 and K3 are the same. The off rates, K minus 2 and K minus 3 are the same. So the equilibrium constants are the same for these reactions. So if you remember back to Ken Blumenthal's talk, he was talking about thermodynamic cycles and Alan Furst. Well, this is the same reasoning. The pathway to from 3 molecules to a complex does not affect the thermodynamics. And so we can take a different pathway. Instead of going this way to get to the trimeric complex, we can go this way. So thermodynamic parameters are independent of the path. So if we were correct in our equilibrium constants for this reaction here, we would predict that K1 equals K4. So the equilibrium constants here should be the same too. So now we want to measure binding of 4E to 4G in the absence or in the presence of cap analog. So for that, we have to use surface plasma and resonance, and this is what the data look like. The 4G is immobilized on the chip. We pass 4E over, and we get on rates from the formation part of the curve, and we get off rates from the unformation, the dissociation part, and can plot those and get a simple one-step reaction. And we find that for either 4E alone or the 4E cap complex, the kinetic constants are the same. So the on rates are the same and the off rates are the same. So the equilibrium constants for that part of the reaction, so this part, are the same as well, 4.5 in both cases. So this confirms what we got from the first time. So why do our results differ? It really comes down to how the 4E was prepared. Now, 4E is a protein that likes to aggregate, and in E. coli, it will form inclusion bodies. So most people studying 4E, that's this recombinant protein, will take these inclusion bodies and put in detergents and sonicate them and solubilize the protein. Or they'll take guanidine or urea and they'll solubilize it and dialyze it out and refold the protein. Every one of the studies that I talked to in Cell and Embo Journal and fancy journals did that. We didn't. So we took protein. So our hypothesis initially was that 4G can dissociate aggregates and produce more 4E monomers that can then bind to the cap. So this made the other authors, our hypothesis, conclude that it's a two-step reaction, that it first binds and then there's a conformational rearrangement and then you get further binding. So increased binding of 4E to the cap has been misinterpreted as an allosteric change. So we tested this hypothesis by making 4E either from soluble protein that had never been in inclusion bodies or by renaturing inclusion bodies. So a test of how native the 4E is how much you can quench fluorescence. You should get about 65% quenching if you have a native protein. And so here we're putting in a saturating amount of M7-GTP. With the native protein, we get great quenching. For the one from inclusion bodies, we don't. Now, I have to emphasize that this protein from inclusion bodies was repurified on a cap column. And so the fact that it is unreactive means that you are capturing the protein on the cap column, but you're also capturing aggregates. So there are other 4E molecules bound to it by multimerization. And this is the protein that all of those other studies were using. So now we do a pull-down experiment with M7-GTP cephorose. What we see with the material from the soluble fraction that we did our studies is that all of the input for E is captured, and you put in 4G and it's also captured. But you're getting, basically all of the 4E captured on the M7-GTP cephorose column. Look what happens with the inclusion bodies. The input is here. You only get about 5% of it bound, and that's what these other papers were reporting as well. When you put in 4G, it increases, so it goes from about 4 to 10%, so you apparently are getting more 4E bound. So this has been interpreted as being an increase in affinity, but it's not. Okay, now I'm going to switch over to the cap analog and how we can modify that. a very useful tool in studying, in producing proteins is to make messenger RNAs in vitro from DNA templates with a cap at the 5' end, because capped messenger RNAs are translated much better, and they can be introduced into cells by RNA transfection, and they're translated maybe 10 to 100 times better in a living cell. So this is done by taking an RNA polymerase, a bacterial or bacteriophage RNA polymerase, all 4 nucleoside triphosphates, and a cap analog, a cap dinucleotide. The RNA polymerase thinks this is GTP and undergoes a condensation here. And so instead of ending up with a triphosphate, you end up with this 7-methyl-G, and so now you make a product that looks like a capped mRNA. And then the next triphosphate will be introduced there, and on it'll go. So this occurs by nucleophilic attack of the 3' hydroxyl on the alpha phosphate, and that's great. You get a 3' to 5' linkage there, right here. But it can also occur by this 3' hydroxyl, and when that happens, the cap is backwards. So in fact, when you do in vitro transcription of RNA with a cap analog, half the molecules are backwards, and these are not translated as capped mRNAs. So we introduced a methyl group at the 3' hydroxyl of the 7-methyl moiety right here, which prevents that from happening, and that forces them to go in the right direction. So we call those anti-reverse cap analogs, because they can only go in the right way. So this doesn't affect cap binding. This is a surface diagram of 4E, and these are the 2' and 3' hydroxyls. They just stick out. So putting a methyl group there doesn't affect binding at all. So in vitro translation experiment, we see uncapped messenger RNA is stimulated by putting a cap on, but then when you put an anti-reverse cap, it's about twice as good. So, and this can be shown in vivo as well. So with these anti-reverse cap analogs that are only oriented in the right way, we attack the problem of mRNA turnover by decapping. So let me divert the discussion for a moment to talk about how mRNAs are turned over. So there are really about 4 pathways. The major one is one in which you have deadenylation followed by 3' to 5' degradation by the exosome, or decapping of the deadenylated molecule and then 5' to 3' degradation. So we were interested in studying mRNA degradation and in blocking one of these pathways so we could study the other, and in particular knowing what's the contribution of the 2 pathways. So we designed cap analogs, or we set out to design cap analogs that could not be decapped. So the decapping, there are actually 2 decapping enzymes. One is called DCP1 and 2. DCP2 is the catalytic, and that's a regulatory subunit, and the other is DCPS. So this is the one that decaps full-length messages, and it decaps by cleaving between the alpha and beta phosphates. This is a scavenger enzyme that at the end of the exosome degradation from 3' to 5', it cleans up these capped oligos. So it has to work on things that are short oligos, and it cleaves between the beta and gamma positions. So the molecules we made are phosphorothioates at either the alpha, beta, or gamma position, and we didn't know which of these would work, if any, but we reasoned that if you go through an intermediate here that requires forming a negative charge here, that this might be more resistant. In fact, phosphothioates are generally resistant to cleavage when they are put into ATP ATP cleaving enzyme reactions. Now, one nice thing about phosphorothioates is they come in two isomeric forms, and they can be resolved by HPLC. So we are just calling these the D1 and the D2 forms for each of these. So they have different configurations. So we express the DCP2 enzyme from E. coli and then did digestion of these capped messenger RNAs with the DCP2. First of all, a conventional cap, that's an ARCA cap, is completely digested to PGPs. That's when the cleavage, well, I don't have one up here, but if it cleaves like that, you'll just get PGP. If you put in a methylene group as a control, it's completely resistant. So we found that the beta thio analog here was completely resistant, but only the D2 isomer. The D1 isomer was partially resistant. So that part of it looked good. Now when we did RNA transfection with HC11 cells, we found a conventional cap, a capped message was degraded rather fast, but an ARCA was degraded slower. You know, half of these are backwards, and so they are very rapidly recognized as aberrant RNAs. The ARCA is more stable, but now the thio compounds are more stable still. We didn't know how they would be translated, but in fact the beta thio compounds are translated even better than a standard ARCA. And I've collected the numbers on this slide. The beta D2 thio is translated 5 times better in RNA transfected cells than a conventional cap. And it is 3 times more stable. So these are really super caps. Now we can go into these cells and start studying the machinery that's involved in degradation without having any decapping going on. Okay, then the last topic is getting into a model system for studying 4E in whole organisms. We're doing this because there are questions about 4E roles. If you remember an earlier slide, I said 4E is involved in mRNA repression, silencing, mRNA transport, but also translation. Studying that in any kind of in vitro system is virtually impossible. You need to have a whole organism, and C. elegans has many favorable properties about this. Good genetics, great genomics, all of the genes are sequenced and many of them are well characterized, a short life cycle. And so we were actually drawn to start studying C. elegans about 10 years ago because there are some interesting types of caps that are found in C. elegans, and we wanted to know what the nature of cap binding was. We were surprised to find that there were 5 different genes encoding 4E, which we named eIF4E1 through 5, and they are all clearly 4Es. They have conserved elements. And that immediately got us thinking, why would an organism need multiple 4Es? It got us thinking about some of these repressors and some of these translators and some of these involved in transport. So we just have started now going through each of the 4Es to understand why we have that number and what do they do. When you look at sequence similarity, so C. elegans was the first, but now we're starting to find multiple 4Es in Arabidopsis and in humans, and actually every eukaryotic except Saccharomyces has got multiple 4Es. So we're up to something like 300 different 4Es. But the interesting thing about the C. elegans proteins is that 3 of them are quite similar in sequence. eIF4E is more similar to human 4E than it is to these other C. elegans 4Es. And eIF4E is up in a class by itself with Arabidopsis and a human protein, and is very different from all of these. So this suggests that the division came early in evolution. The approach we've taken, remembering that 4E is a translation factor, if we wanted to study the role of a particular one in this organism, we would like to know how does it affect translation. So the approach we took was to generate a genomic knockout and then look at what proteins were affected in their expression. So when you knock out eIF4, there's a complex phenotype, egg retention, low brood size. So you have a delay in eggs being laid. You can restore that by expressing the eIF4. There's also hypersensitivity to serotonin, and so a complex phenotype. The approach we took was to do translational profiling, and the idea here is that we take polysomes and look at the high and low regions of the polysomes, comparing wild type and mutant. So we take the light polysomes and do an Affymetrix DNA array experiment, and the heavy polysomes, and then the total, and we ask which messages are underrepresented in the mutant in the high or the low region, and which ones are transferred up to just the untranslated region. So by comparing all 18,000 genes in C. elegans, we can see which ones are the most affected. So here is looking at mutant versus wild type. Most messages are not affected in high or low or total. Some are overexpressed, most are underexpressed. Most that change are underexpressed in the mutant compared with the wild type. And when we start looking at the identities of those, these are ones that are prominently changed. So this means that there's a threefold loss from high polysomes of this message and a threefold loss in low polysomes. So it's being translated poorly. And these, they're called eggles because their phenotype is egg-laying. When you mutate that gene, they don't lay eggs very well. And that's the same phenotype we're getting. So we can show that at least some of the phenotypes can be explained by underexpression of these proteins. We went on to look at the expression of these proteins, and they are, in fact, underexpressed, but I won't show that. So another way of looking at the most affected genes is to plot the low versus the high polysomes, and then the signal log ratio, so log 2 ratio. So these are the ones that are decreased in high and low polysomes. So this quadrant right here represents about 300 of the genes compared with overall about 10,000. And so we now wanted to know what is common to all of these genes. Why are these affected by the loss of eIF4, whereas all the rest of them are not? So this is where we did a bioinformatics approach. From public databases, you can look at the developmental expression pattern of all messenger RNAs in C. elegans. And they can be grouped with a self-organizing map into 25 different groups. So this is just the ones that start low in embryos, and they increase during larval development. These increase up to, and they peak in the 4th larval stage and start coming down. So you see all patterns. These decrease from embryos to adults. So those are 25 patterns, and we gave them all different colors. You can then look at the polysome behavior, the same plot I just showed, and color them as well. And we see that the ones that are kind of purple have more messages that are in this quadrant here. So they're the most affected. And so you can see that when you plot them all in the same one. The purple ones really bias towards this quadrant of the plot. So that means that messages most affected by eIF4 are the ones that have this developmental expression pattern. They peak at L4 and then they come down. So this, in fact, is where eIF4 peaks. So now we look at expression of the eIF4 protein itself. It peaks at L3 and then starts to come down. So this is at least part of the explanation for why those messages are affected primarily. Okay, I am done with my time. These are the people that did the work. Cece did most of the C. elegans work. Cece Dinkova, Eva Grutzian did the cap analog work. Sergey and Nadia did the biophysics work, and the others were working on other projects. So thank you very much.